Okay, hello everybody. Good afternoon and welcome to the Health Research Institute seminar for February. Sorry. So I want to begin today by acknowledging that we're on the traditional unceded territory of the Kwajalein. And just if we can all just take a minute and reflect on really what that means for our lives personally, living in these beautiful lands. So this is your HRI seminar series committee that's organizing the seminars throughout this year. And if you don't know me, myself, I'm Dr. Kendra Ferber. We also have Dr. Rihanna Robinson. Nuzia is our student representative, and of course, Liana and Bree from the HRI that do all the work for us. And we have a bunch of contributors and sponsors to the seminar series, departments across UNBC, as well as Financial Life. So today we have Dr. Clindy Morgan presenting, but just a reminder of our upcoming presentations, there's only 2 more. In March, we have Dr. Linda O'Neill here. And then in April, I hope you all can join us. We have our honor students, the health science honor students presenting their projects for the year. So Dr. Clindy Morgan joined UNBC in 2021. She has a PhD in chemistry from the University of British Columbia under the supervisions of Dr. Raymond Anderson and Catherine Ryan. So she has conducted, her research expertise is in investigating bioactive natural products from marine and terrestrial bacteria. This also includes innovative work in developing novel methodologies such as nitrogen-15 NMR for natural product discovery. So Dr. Morgan's research program is currently funded by NSERC, where she looks at antifungal compounds for plants. But today she's going to talk to us a little bit more about the potential applications for human health. So thank you very much for joining us.
I'm sharing the share.
Sorry, that was my job. I told you.
You did okay at the top as well.
And then I
go to PowerPoint. The natural products.
And then
I'm going to the slideshow. All right.
No, and then we just have the share screen.
Can you share the screen on the screen there, Clindy?
Oh,
do you want me to do that?
Did exit?
Exit first.
Oh, and then there we go.
Cheers. You can move that. We need that out.
No, yeah. Okay. And then do you want presenters down to the bottom? Perfect.
We got it. You think we know how to do that by now?
All right. So as Kendra mentioned, so far my research program here at UNBC has been focused more on agrochemical discovery, although I'm starting to venture into human-focused discovery. And so due to that, this talk will focus on a broader interest of natural products and how they can, why they're so useful, where they come from, and why they're so useful for pharmaceuticals. And so I'm going to use case studies from the literature to talk about that, and then end with some of my own research that a little bit might be a little bit of a tangent, but hopefully you can see the overlap. All right. So, I guess I can't move. Oh, I can. Okay. All right. And oh, no, it doesn't want me to do that, but let me see. Okay, there we go. So natural products, a lot of people, it's a very common term actually. It's used quite broadly in society. It can be thought of as things that you go to the health food store and purchase. But this, in fact, when I'm talking about natural product chemistry and their uses as pharmaceuticals, this definition is too broad and not exactly helpful. So for those who study the chemistry of natural products, tends to be natural product chemists and medicinal chemists and others, really, we tend to be discussing a particular set of compounds that are derived from nature that are termed secondary or specialized metabolites. We'll be talking more about this, but in general, what secondary metabolites are, they're small organic molecules. They're marked by an intense amount of chemical diversity and complexity. They're studded with heteroatoms and multiple stereocenters, and they're produced by living organisms for defense and communication. All right. So we are all actually quite familiar with natural products in our lives. Although it's probably only been over the last 200 years that we've understood the chemistry behind the activity, the reality is that probably for as long as humans have been interacting with plants, we've been interacting with these secondary metabolite natural products. So as an example, here is frankincense, which is obtained from the frankincense tree. And that smell, this is actually just a small subset of the secondary metabolites, the specialized metabolites that frankincense produces, and that is associated with the very potent and sacred scent of frankincense. And they work together to produce what we associate with that scent. Some other ones, limonene, and again, this is a very small terpene that is produced by a variety of plants, something we're very familiar with, and oftentimes it will be mingled, say juniper, caraway, or sage, it'll be mingled with other chemicals to produce the scents that we associate with those plants. And same, linalool and nerol, again, small terpenes. So this, we're very familiar with the scent of terpenes in our lives and the taste of terpenes. They're in cardamom, lavender, bitter orange, and wine, for example. However, while I want to focus on the fact that these are potent and beneficial chemicals that are coming from plants, it's really important to realize that plants, just because it's from nature, doesn't mean it's harmonious. And plants are also prolific producers of very toxic compounds that can kill us with just small amounts. So we have the hyoscyamin from belladonna. We have we have potatoes, which produce a solanine, which can kill us. And we also have, this is a little compound from comfrey that can really damage our liver. But of course, it's also cytotoxic. So it's being explored as an anti-cancer drug. So it's all dose dependent. All right. So that's natural products in our everyday life and the fact that we actually have an intimate association with them. But one of the things that makes them so useful for pharmaceuticals is their structural diversity. So I'm just going to show a subset of the compounds. So this compound is actually produced from valerian. And it is the major, when they started to actually, people have been using valerian as a folkloric medicine for sleep and headaches for a long time. When they started to investigate the chemistry behind that activity and doing something called bioassay-guided fractionation, what they found is that this compound here is actually the major component of those active fractions, but it's not very soluble in water. And so it couldn't be giving the activity if people are drinking it in tea, but yet there's that common anecdotal evidence of the fact that valerian tea gives us sleepy effects. And so they actually found valeric acid, which was 0.3% of the active fraction that they believe, and further tests show that that is actually what produces that active, the sleepy effect. And so this shows how important it is for us to actually understand the chemistry in order to understand why plants or other compounds might be of use to us as humans. These are a series of chemicals. They come, permethrin comes from chrysanthemum. It's a natural insecticide. People have used chrysanthemums for generations as a natural insecticide, but now we can understand the chemistry and where it comes from. And hopefully too, we start to see that there's this pattern of very diverse structures. Then we get this rare complex tetraterpenoid from the bark of a fir of Tibet. And this is a very interesting compound. It's synthesized, or it's the biogenesis is from a Diels-Alder reaction. And again, I point that out for those who might remember their second year organic chemistry, is that it's the structures and it's the structures that dictate how these molecules come together. It doesn't matter the source, whether you're doing it in a laboratory or whether you're doing it inside of an organism. It's the structures that will dictate how the pieces come together. And people were very shocked that there was Diels-Alderase enzymes out there, but it makes perfect sense when you think about the fact that the chemistry dictates this. All right, we also have lovastatin, another compound coming from a Diels-Alder for its biogenesis. And it's one of various phagocytostatin. A lot of people take this drug, and it comes from filamentous fungus. So all of these are from plants, and we're starting to see some of the structural diversity that's coming from other organisms. We also have two very important pharmaceuticals that come from Streptomyces. This one is found from a Missouri soil in 1945. So Streptomyces are a bacteria that are fascinating bacteria. They are the most prolific producers of natural products of any bacterial clade. So this is a pharmaceutical, and this is another nystatin, which you probably recognize. It's in products that you find over the counter. And again, showing the intense structural diversity that we can see from secondary metabolites. This is a marine invertebrate natural product. No, I will be talking about some marine invertebrate natural products that have become pharmaceuticals. This one is just one that's very interesting structure, and that was isolated and the structure solved. All right. So again, moving into our modern uses of natural products, and in fact, a large majority, especially of our antimicrobial and our anti-cancer pharmaceuticals, come from natural products, either extracted, semi-synthesized from those natural products, or synthesized. And what I mean by that is their scaffold was changed in some way to get that pharmaceutical, but the originating, the origin of most of the structure comes from the natural product. And then we have a very small percent, actually, that is derived through the minds of medicinal chemists or other pharmacologists. Okay. Also, this is, I probably should take this up, actually. This is my own personal interest. So this is agrochemicals. Right now, most of them are not natural products. But anyway, I'm going to skip that because I should have taken it out, sorry. All right. So in terms of historical natural products, and again, the overall thrust of this is that we're very familiar with them. They have been in use as medicines for a long time, and their structures are really interesting. So in terms of the history of natural products, morphine and strychnine were two very early compounds isolated in there about 200 years ago. They're very complex, and their structures were actually not solved for 100 years. These are very complex compounds. People have been working on their synthesis, but in terms of actually producing enough to be used the way that we use it for medicines, we still extract them from poppies. There has been some attempts at biotechnology in putting these gene clusters in yeast, but actually, although that happened now about a decade ago for the first time, there's the barriers to actually making it a viable system for production of morphine as a medicine has not yet occurred. So it's still extraction from plants. Then we have penicillin. And this really started the microbial natural product revolution. So just about 100 years ago now. And its structure was also not solved for about, just a second, I think it was 40 years or 20 years. I'm getting myself a little bit mixed up here, but yeah, just look at that guy. Yeah. 20 years. In the meantime, they were doing large-scale extraction from fungal cultures for the use of the penicillin penicillin as an antibiotic in World War II. Once they solved the structure in the late 1940s, then they were able to work on synthesis, and they solved its synthesis. They did the first photosynthesis 10 years after in the late 1950s. This penicillin G tends to be produced for pharmaceutical use through synthesis. So, and natural products are inherently interdisciplinary. They are produced from biology, but it's the chemistry that gives us the activity. There are many, many fields that work with natural products and where natural products has advanced those fields in deep ways. So as a natural product chemist, I'm interested, I'll talk about that. So we have chemical ecology, microbiology, biochemistry, chemical biology, bioinformatics, synthetic biology, and bioengineering, pharmacology, all which can be centered around natural products. So for myself as a natural product chemist, this is a subdiscipline of organic compounds. And it is a study of these organic compounds, which generally are under 2,000 grams per moles, or what's called small molecules. They are produced by living organisms, but do not have an essential role in the growth and repair or physiology of the organism. And so we typically study the separation, the structure determination, the biological reactivity, semisynthesis, biosynthesis, and synthesis of these structurally complex molecules. Really, and we're very interested in discovery of novel scaffolds in the chemical space, the bio. All right. So I titled the talk Nature's Defense, Nature's Defense and going towards pharmaceuticals of strength. So for myself, it's really important to understand that these are and how they're produced and where they come from and why. And so if I say that they're not involved in physiology, growth, and repair, why are they made? They're actually extremely expensive for the organisms to make them due to their structural complexity. They tend to have extremely large specific gene clusters that are that are required to make these compounds. And as, for instance, something like Streptomyces, they might be producing 14 to 15 different natural product scaffolds and then have a diversity of structures within those scaffolds. So they can make quite a few of them, and some of them don't actually have uses because the whole point is to make structural diversity in case it has uses at times. So again, it's this niche adaptation in ecology. They're often going to be mediating the interactions between the microbes of a plant's rhizome and the plant itself. This has a lot of evidence behind it. We know that they mediate interactions between pollinators and flowers. So in this case of chemical mimicry, the wax of this orchid produces the hydrocarbons that the female bee produces, and they actually use this to attract the bee for pollination. And we also have, again, we have them, there is 375 novel natural products that most of them antimicrobial, many of them antifungal, that have been isolated from bacteria and fungi that live with termites and mediate their relationships with the trees and the other fungus around them, particularly the pathogenic fungus. So these are some examples of the relationships that these natural products are mediating. And so because of the fact that they're all about ecology and niche adaptation, strains of plants in different areas of the world can be producing different natural products. Strains of bacteria, even isolated from the same cubic meter of soil, can be producing different natural products because the whole focus, they have this evolutionary drive to produce complexity. And so their mechanisms have evolved that favor the production of this chemodiversity. And so the whole biogenesis of these natural products occurs in a diversity-oriented manner. There's promiscuous enzymes that have relaxed substrate specificity that diverges them from primary metabolites where there tends to be stringent substrate specificity. They have modular gene clusters for production that are amiable to the gain and loss of particular genes within those gene clusters that will end in the changing of the chemical structure. They have genomic placement that leads to greater numbers of mutations, that leads to greater numbers of chemical diversity, and there's even more mechanisms for this. And in fact, this last one, there was some evidence for it. It was more of a hypothesis, but there was just this one paper where they sequenced the whole genomes of 1,000 bacteria and were able to show conclusively that this is true. So as a summary of everything that I've been talking about, so far, natural products are genetically encoded. They are subject to evolutionary optimization. They provide niche adaptation, they have ecological roles, and they influence inter and intra-species interactions, including defense and communication. And biological diversity really drives the chemical diversity. And what this means is that we can utilize and harness unexplored biological niches to yield new chemistry. And so the consequences for drug discovery of all of the push for chemical complexity is that they are rich in stereogenic centers. They often have multiple quaternary carbon centers. They have adjoined aromatic or aliphatic polycyclic systems, and they have complex defined three-dimensional structures. And they have usually a very high oxygen and nitrogen ratio to carbon. And it's that oxygen and nitrogen that really functionalizes our organic molecules. So again, they're primed for bioactivity. And one of the aspects of these structures that really sets them apart from those chemicals that are invented from the minds of a medicinal or synthetic chemist is that it's the variety of substructures which endow the natural products with more complex three-dimensional typologies. And then with these well-defined three-dimensional typographies, there's an entropic benefit that comes when the drug interacts with its target. This helps contribute to its overall use as a pharmaceutical. Probably why so many of our pharmaceuticals come from natural products. So again, chemical space in and of itself is huge. And I'm talking about organic chemical space, where we can intermingle our carbons and our nitrogens, our oxygens, sulfurs, to a wide, it's probably we could have structures that are as countless as there are stars in the sky. And in fact, the chemical space that natural products occupy is also enormous, yet it's filtered through the need to interact with other biological organisms. And this is why, this is partly why they bind and interfere with a much wider range of biological targets, why we can potentially use them to discover new mechanisms of actions, and continually, if we can find new structures, we can potentially we can solve part of our antibiotic resistance issue. Now I'm going to switch focus from discovery to drug development. Well, I'll be talking about some case studies that will be going through the discovery process and taking us all the way to the pharmaceuticals, and actually show that this timeline can be quite long. All right, so one, it is a little bit biased to some marine invertebrates, because I come from a lab that studied a lot of the natural products from marine invertebrates. However, I should say that one of the very interesting things that has come about with the genomic revolution is the understanding that many of our most complex and interesting compounds are actually likely produced by microorganisms, and particularly bacteria, that are in symbiotic relationships with the sponges and other animals. All right, so this is the natural product Helicondrin B. And this is halivin or eribulin, which was derived from Helicondra B over many years. So so as halivin, it was approved by the FDA in 2010 as a drug to treat patients with metastatic breast cancer. And it did increase longevity of those who took the drug. And it did have some side effects, although I believe that's normal. And the price is it's not cheap. Okay. But from the discovery itself, so it was isolated with 7 other helicondrins in 1986. So 2010, it was approved by the FDA, and it was first originally isolated in 1986. So that's a very long period of time. Helicondrin B was also found in other species of tunicates. But this was this first foray into these structures. I'm also giving a little bit of background into what's called bioactivity guided fractionation, which is a huge component of how we discover new molecules, but then 30 years later or so can get turned into pharmaceuticals. So this is the scheme from the paper itself. And this is just a little bit of more of a rough guide through. So usually we assay our crude material whether it's been an extract from a marine invertebrate, so you soak them in some sort of organic solvent, or if you're doing bacteria or fungi, same thing. And then you begin separating those organic compounds into pure fractions using tools that might be familiar from organic chemistry labs, such as silica flash chromatography. Then you usually monitor that purification through and the chemical structures through things like TLC, NMR, and mass spec. And you also monitor each round of fractionation with the bioassay. So that is why it is called bioactivity guided fractionation, because you're using the results in your bioassay. As you start from one crude and you start multiplying and getting many, many fractions, you're using the bioactivity to help you select which one you take on to further separation. And this also gives you a little tool. Since you're starting from a complex of material with, if you take out, say, 40 micrograms of that complex of crude material, only a small percentage of your active component will be in there. But as you fractionate, your active component should be increasing in concentration. And so this actually gives you a handle to see. So as you separate, your potency should be increasing in your bioassay. And this helps you know if you have a false positive or not. Once you've been through multiple rounds, we can see here how many rounds they went through. And sometimes you have to try one thing and it doesn't work, and you got to try something else. And so by the end, when you get down to it, you usually go to a more refined technique like HPLC, which has a much higher resolution. Apologies, if a high pressure liquid chromatography, for those who don't know, and you obtain a final pure compound. This can take a great deal of time. Then you're normally very material limited, particularly if you're working with marine invertebrates. And so you solve the structures with non-destructive techniques as much as possible. Again, that is assuming some knowledge, but, and then once you have the structure in hand, then you will test it again in an assay to determine if your initial bioactivity was in fact true for the pure structure. And this is just the first part of isolation, right? So this might take you a couple of years depending on the compound. So this is a structural elucidation from 1986. So everything is likely hand-drawn instead of now we would use a software program, but these are the techniques that they use to solve the structure. And then they began to analyze the bioactivity. And in this case, it was really lucky because, as I mentioned, they isolated it with a number of natural analogs. And the differences in the bioactivity of the natural analogs started to give them a clue as to what parts of the structures might be important for bioactivity. But it was produced in extremely tiny amounts. And when they isolated it, and you need about 10 grams of material for preclinical development. Sorry about that. And so if it is approved, you need somewhere on the order of kgs. You cannot, with marine invertebrates, you cannot do that through extraction because you have to go down and you have to dive and you have to harvest the marine invertebrates. And if you were to do this much necessary a year, you could end up killing off your species. So there's solutions, which is often gone to. One is total synthesis. And this was really important in this particular natural product. And there also was a concerted effort to actually do some deep sea diving. So go to the natural source. There was some, and sometimes people worked on doing aquaculture where they actually grow the marine invertebrate in different cases. Okay. So while they were doing their total synthesis, they were also working towards what's called structure-activity relationship studies. So they'll synthesize parts of a molecule, subject those to the bioassay and see which parts of those molecules are important for the bioactivity. And I'm not showing the total synthesis, it was many, many steps. But what this resulted in is that they found out that the right half of it was actually the most important part for the bioactivity. This was really helpful. They didn't have to worry about the rest of this. So the SAR testing allowed simplification of the molecule. And so this was the halocondrin B, the natural product, and this right half was a synthetic intermediate. And in fact, it was actually more potent than halocondrin B. So this is the bioactivity. They found that it was tubulin depolymizer So it's complicated a little bit. Probably some of you in the audience might understand this more than I do, but this is the overall way that it interferes with tubulin production. So again, this is now that they knew that only the right-hand side was important in the activity. Now they could focus on a more optimized synthesis of the right hand, and this led them to eribulin mesylate, and this provided enough material for clinical trials. And this is how there's probably been some optimization, but it's through total synthesis that this drug is produced for market. So again, long timeline, 1986 to 2010, for this was first isolated and going through clinical trials to be approved at the FDA. So I'm going to talk about another Cedrus from Dolastatin 10. This is from a sea slug. And this was, in fact, now with the actual compound, the actual pharmaceutical, it actually got more complex than the natural product. This is the part here that's derived from the natural product, and they added some additional bits that were necessary for pharmaceutical activity. So this was much shorter. This was only a 15-year effort, but it was first isolated in 1981. And from 100 kilograms of sea slug, wet sea slug. So water contributes towards that weight. And so once they isolated and solved the structure of Dolastatin 10, they had 29 milligrams of this compound. So this is often the hardest part of natural product discovery. But it had greater bioactivity than pharmaceuticals in production, so it had interest. And so here in 2001, they isolated this from the marine cyanobacteria. So again, showing, this was the start of the story of realizing that sometimes some of these compounds are being made from this bacterial symbiont, which then opens up things like fermentation and other ways of accessing these compounds. And this is the bioacid guided fractionation. So soaking your sea slugs in a mixture, in this case, of ethanol and isopropanol, and then doing a various solvent separation, and then until you start getting into your chromatographic separations, and then your pure compound. And this was, it took quite a while. The synthesis was not, the structuralization was not trivial. They had to do a lot of, they had to do hydrolysis of the peptide and compare it to known compounds. And it was quite, because there was very unusual amino acids in here, which marks a lot of our natural products. Finally, though, they were able to get the correct structure and sequence of the natural product. So this is just showing how they used proton NMR, carbon NMR, and NOEs to solve the structure. And some of this might not be familiar to some of you, but these are very typical methods that we use to solve structures. And then, of course, so the absolute configuration still was not known. And so they had to synthesize it in order to get that structure. So this is the scheme of the synthesis of one of the parts of the, one part of the natural product. A lot had to go through in order to get these unique amino acids. And this is the synthesis of the other part of the peptide, until they finally had the full structure. Just another scheme here. All right, so once they had that full structure in place, then they were ready to, they produced a whole lot of it. And again, it too was also a tubulin disruptor, and is used to treat cancer. And this is some structure activity relationship studies where they were able to, so they took out some of the more complex aspects of the structure because they could simplify it, and it showed that they would still get similar bioactivity or better. Okay. So this was the natural product that was isolated and solved. And then this is the component that comes from the natural product. And we can see that they've simplified some aspects of this structure. All right. So the third one that I will talk about is caspofungin. Okay. So this came from now a fungal a microfungi, so called Glariera losiensis. So it was isolated. So this is the natural product, pneumocandin B0, that was isolated from 10 liters of solid fermentation. And again, through bioassay guided fractionation technique. So red is where they found the bioactivity. And so, for instance, the hexane would be not active and they would dispose of it. Oh, sorry. Oops. I copied that. That should not read dalsatin 10. That should read pneumocandin B0. Copy paste errors are my absolute favorite. Okay. So this, in fact, so with pneumocandin B0, it actually, the overall structure did not need to be changed much. There was only a couple of aspects. There was some issues of solubility. And then this little ornithine derivative here with the dihydroxylation was proved to leads to instability and led to cleavage of the molecule and would inactivate it. So they substituted that with an amine, and that solved most of the issues. So in fact, caspofungin is produced through a semi-synthesis strategy. Pneumocandin is isolated from large-scale fermentation of the fungi, and then there's just some small semi-synthesis that's done to finish off the molecule to prepare it for its use as an antifungal. And so these are 3 of our most important antifungals. And likely because of the didn't need to change very much of the structures, it actually took 11 years from initial discovery to FDA approval. And these 3, they're all relatives of each other. And they are, in fact, our first-line treatment for a variety of fungal infections. And there has been a couple, people are working on their diversifications. So far, there hasn't been much in the way of resistance. However, there's been some first signs of resistance towards these antifungals. So diversification of the structures is something that is of interest. And yes, worth $500 million in the market if one is thinks about those things. All right. Okay. So the takeaway from this aspect of my talk. So the natural products as pharmaceuticals. So they continue to be important sources of diverse drugs with novel targets. Like all pharmaceutical development, it takes rigorous and expensive research and development. And this is a really important part of using science to explore these natural products. However, we want to interface with natural products, it is very important that we understand the chemistry behind them because chemical structures underlie all the activity. And plants, fungi, and bacteria, they house potent chemicals that can kill us or benefit us. So again, you must treat them with respect. I mean, probably going to be treating bacteria with respect because we know that they can be pathogens and kill us if we take the wrong one. But particularly with plants, we need to be mindful of the fact that it's not an invisible or magical source of why we can use them for our health. It comes from the chemistry. And again, it's dose dependent. Right. So now I will switch gears to my personal developing research. I have been initially interested in investigating the chemistry of the northern boreal insect-associated bacteria towards antifungal and sustainable agrochemical discovery. So initially I started pursuing this for agrochemical discovery, but it appears through both genomic and some initial assays that there are a variety of antifungals, particularly the genomic genome sequencing has showed me that there's quite a few antifungal compounds that are potential in there. So I think that it's we'll start pursuing these as a source of human antifungals as well. And the reason for the northern boreal insect-associated bacteria, of course, is those novel niches. All right. So I'm also looking at the isolation, semi-synthesis, and synthesis of a genetically encoded chemistry. This is tied into my insert grant. And again, the reason for this is because one of, so as I mentioned in the about 100 years ago, penicillin or the assay or the phenotype of penicillin, of the Aspergillus, which produces penicillin, showed us that microbes can be a potential source for pharmaceuticals took, right, 30 years before we understood, or 20 years before we understood the chemistry. But since that time, from the 1950s until around 2000, there was massive efforts of microbial discovery by drug companies like Merck. But what ended up happening is that they ended up doing a large amount of rediscovery of the same compounds. And since it can take a huge amount of effort to isolate and solve the structure of a compound, this was diminishing returns. And they said, this is not worth it. Let's move away and let's start going into more of that. Let's design and devise drugs, these large libraries with high throughput structures. But they found that there, in fact, even with the case of discovery, not rediscovery, natural products are still tend to be a better source of structures, particularly for antimicrobial, anti-cancer. And then the genomic revolution happened. And with the genomic revolution, we can look into the genomes of our organisms, particularly bacteria, and we can predict what compounds they're going to be making and if those compounds have potential novelty. And that is that big fact that we can predict if they have potential novelty is really important, because that means we're not going to waste our isolation effort. So we know that we're not going to get to the end or for whatever reason and get to a known structure. I mean, it's not terrible, but it's like can be quite expensive. There's a lot of solvents that goes into that process. So we want to stack the deck in our favor. And then working towards flow chemistry and other production of vitamins and natural products, and towards this effort, again, to efforts from my perspective that have great returns for our effort. We're looking at using ideally working towards using some of the biosynthetic genes that make these natural products and turn those into biocatalysts. Of course, doing that with collaborators who specialize in enzymes. So again, so we have this underexplored niche for antifungal natural product discovery. So this links back to the chemical ecology, or the 375 novel natural products isolated from bacteria and fungi from termite associations. And so, in fact, in our forests, we do have this up here in northern BC and the Yukon and Alberta, we do have a number of species of bark beetles. And aside from those bark beetles, we have a number of other insects, but bark beetles in particular, we all know the story that they have this deep symbiosis with fungi that helps them overcome tree defenses and cause widespread destruction of our forests. Well, they also have bacteria that live within those symbioses, but the chemistry of the bacteria has been largely unexplored until now. All right, so as mentioned, the bark beetles have associations, the tree hosts, the fungi, including yeast, mites, nematodes, and bacteria. And there's a lot of research into these symbioses that's ongoing here at UNBC. It's a great interest. All right. So, but it's these for myself, coming from the view of bacteria. It's those bacteria that are very interesting. And we know in other similar systems that are producing novel, useful compounds, and particularly antifungals. So hopefully, again, stacking the deck in our favor, my favor, of finding new chemistry that's useful as antifungals. So in order to do this, it's a step-by-step process. So we actually have to go and gather the bugs and the galleries from the trees where the bugs live. We have to isolate the bacteria and then store the bacteria because we can't deal with them all at the same time, and then come back to it and start exploring the chemistry. So this is the part that's been involved in for the last few years with a lot of help from a lot of different students, which we'll talk about at the end. And also help from other professors at the university and other Ministry of Forests in the Yukon. So this is just a subset of collections that we have done, or I have done, and with help. So with the Hoover traps at UNBC, we collected Douglas fir beetles, and then went up to the Yukon. The Ministry of Forests sets up these traps. It's actually really, and I should say this is on the Champagne and Ajax First Nations land, and it was they allowed me to obtain these beetles from the Ministry of Forests in order to do some scientific research. So thank you for everyone who helped me out with this. But this is really interesting. I found this out when I went up there. So in fact, most of the heat of the Yukon comes from wood. What it comes from is beetle kill wood. So we monitor beetles here in BC because we want to make sure that we are not having infestations of beetles. They monitor beetles in the Yukon because they're trying to predict where their next crop of dead trees is going to come from to provide half the heat for their 40,000 odd citizens. So this is just this yearly endeavor that they do in order to find out where they can, and then it's local people who go and cut down the trees, and that's part of their income as well. So it's a really interesting economic system. So they set these up. This is a trap near the Kluane National Park near Haines Junction. In the Yukon, and I went with them, and we had to travel quite a few different distances, and we collected northern spruce engraver beetles as well as spruce bark beetles. This is their Ministry of Forestry truck. So they had to count them for their records, and then they let me keep the rest. This is a student back several years ago when we were first developing so we get the beetles, we take them back to the lab, we crush them, and then we start plating them onto what we call diversity plates using different media. And we're particularly interested in Streptomyces because we know there's been a lot of research that's now shown that despite the fact that there's a lot of really interesting compounds from Streptomyces, we know that there's a huge potential for still more chemistry to arise from them. So they can take a long time. They can take up to 8 weeks to pop out, very slow growing. They can get easily outcompeted by the faster growing bacteria. So we leave these plates and we store them for a long time. We periodically check them. Okay, and then this is just there's obviously some molds and stuff there, but then we pick off the little single isolates that are in our diversity plates and we isolate them until we subculture them to purity. Okay, and then once we have them isolated, then we store them, and then we start subjecting them to plant pathogen bioassays. We will be subjecting them to human pathogen assays. And we also, not shown here, but we also do spectroscopic studies. And so the goal, of course, though we're just beginning our path, the goal is to isolate novel structures that can be useful potentially as phytopathogen assays. Potentially, we can understand more about the relationship of any of this chemistry in the symbiosis of the bark beetles and the trees. That also done with collaboration, and then some human health impacts as well. Okay. This is just some of our bioassays against some of these phytopathogens. Okay. And then once we have our bacteria, then we start growing them and doing the chemical analysis, organics extracts. And again, this is just the beginning. I decided not to show some of the more complicated data just because, but we have our TLC and we have our different colors. This is a really interesting compound that's being produced by this bacteria. It's actually a pH indicator. So it will be green when it's above a certain pH, and it'll be red when it's below a certain pH. And then the other aspect that we're working towards is the flow, and also just beginning is the flow chemistry production of vitamins and natural products. And part of this is developing biocatalysts in projects with Dr. Andrea Garrell, who is an enzymologist. So we're working on the flow synthesis of vitamins. And then a project that I am keen to start is using what we know about some of our more interesting and diverse structures and their associations with bioactivities, and then helping to diversify some of our known antifungals to potentially provide the tools for future antimicrobial resistance. So for example, this is piperazzic acid. So piperazzic acid is a non-proteogenic amino acid that is produced for use in secondary metabolite peptides, which are different than primary metabolite peptides because they're all about structural diversity. You have all of these hundreds and hundreds and hundreds of interesting amino acids that are produced specifically for incorporation into these secondary metabolites. And this one is a 6-membered ring, hydrogen bond. And there's probably now about 150 compounds that have been isolated that carry this structure. And of those, a large percentage have displayed some sort of bioactivity. They tend to be produced in smaller amounts, maybe because of that or other reasons. None of them have yet made it through to use as a pharmaceutical, except for this natural product inspired design. So they took boracic acid and then they used medicinal chemistry tech modeling to produce this, which is now a pharmaceutical. I would like to use PIZ in this similar way. So it is produced by a very specific clade of bacteria. And so these are caspofungins, these are antifungals, and they're produced by fungi. And so they would not have access to PIZ and its particular, so it is a proline analog. So like proline, it provides a lot of structural rigidity. However, when bound into a peptide or when covalently bound into a peptide, we still maintain a nitrogen-hydrogen bond because one is available. And so my goal is to potentially produce these analogs with PIZ incorporated into these caspofungin and see if we can provide alternate methods of, well, increase our structural diversity and perhaps even increase our solubility and availability in the bottom. Okay, so to end, just thank you to all my students who've been working with me for the last few years and very patient. All right, so I've had a number of students isolating the bacteria and helping to freeze them. So starting in the summer of 2022 and into this last summer. And many came from Mitacs. Very grateful to Mitacs. Really, really amazing opportunity. And we've also had master students and a student in the audience who were exploring the bioactivity through bioassays. And then I've had a number of students exploring the chemistry to different degrees, whether it's genetically encoded bacteria or compounds and trying to semi-synthesize them, or whether it's starting to explore the chemistry from our bacteria. And more will be coming because this is now our large effort. Okay. And then have a number of students as well who've been working on the flow chemistry, those who are working on the synthesis aspects, and Annika, who's co-supervised with Dr. Gorell, who's working on the biocatalyst, and Andrea has also had a few other students who've been focused on this biocatalyst development. All right, so thank you for listening. Any questions?
I have a question.
Yes.
I don't know if you already told us, but when you have the beetles collected and then you grow them on the petri dish, is it the beetle itself or you use the larvae?
We use both beetles, and the larvae are harder to gather. Okay. But I actually found some infected trees when I was hiking, and so we collected some of those. It has to be in the winter time. So the larvae are in the winter, and then they evolve into adults in the summertime. Yeah. Thank you.
It's hopefully on that side. You do not foresee yourself needing hundreds of kilograms of beetles, correct?
No, because we're looking at the bacteria. We actually kill the beetles, which is probably a good thing. We had a few extra beetles. We're like, I don't know if we should just dispose of these or not. Like wood pieces of beetles are like, oh, yeah, don't want to let those out into the environment. So.
Can you explain a little bit more about what you said about why these secondary metabolites have a relaxed interaction structure, like why they can interact with
multiple substrates?
Yeah.
Well, I can't say exactly why. I don't even know if that's been solved. There's people who have hypotheses of how that comes to be, but the overwhelming evidence is that that you can test those enzymes. And in fact, the RINE does that because they actually solve the structures of enzymes, of these biosynthetic enzymes. And do the purification and all that. So yeah, they will routinely test them with the different substrates and they will accept. There'll be varying levels of turnover, but yes, they will accept them. Does that answer your question? Probably not to some degree, but yeah, it's hard to say why, right? Yeah, I mean, again, the push would be the fact that you're producing more compounds. That like I said, there's some few hypotheses of how that evolved. They would have obviously evolved from the primary metabolite enzymes. But then, I mean, it's like, why did our eyes develop? I mean, like it's kind of like it's a broad question.
Yeah, I was just wondering if there's some particular characteristic about like these metabolites that they're particularly flexible or.
I don't think they're flexible. I would say probably the active sites. Yes, or the active sites would have to be potentially looser. I would imagine, because you want to be able to catalyze that reaction, even if you've changed the structure that's in there a little bit.
Thank you for your presentation. I was just wondering, I did some reading a little while back about microbial derived metabolites, and I might butcher this name, but I think it was called fulvic acids or something of that nature. And I was just curious if you've come across that at all.
I haven't, but
okay.
Was there a particular use?
No, it was honestly, the interest was just because there's a growing interest in, bacteria and, even in the ecosystems of our gut and so forth. They were saying, well, it's complicated, so maybe we can look at bacterial byproducts. And then I think it was, I'm not 100% sure, but.
Okay, because that is a whole subsection, yes, of our human microbiome, which are mediated in part by natural products, but they tend to be simpler.
Okay.
In structure.
Okay.
That's
cool.
my understanding from what I've read about that. Yes. Yeah.
Thank you.
Thanks so much. I'm really excited to hear about this work happening on campus, and that you're and I really appreciate you coming to this seminar series. So thanks for that. I'm really curious, and you might have sort of alluded to this, but a lot of the kind of development that you were describing is sort of anti something, in the sense of it's, you're looking for products or chemicals that could be antifungal or antibacterial or anti-cancer, which is, as somebody with a medical background, that makes perfect sense. There's things that we don't want anymore. We don't want pathogens, and we would prefer not to have cancer. So that makes sense. But I just wonder, of the proportion, like, it's probably a really hard question, and it might be sort of beyond your area, but like of the proportions of natural product development that are happening out there, where would you say that the big emphasis is? Is it true that they would be mostly sort of anti-pathogen and anti-cancer, and then there would be some other things that are, I guess sometimes there's other products that people are interested in because of their health promoting possibilities?
Yes, and so it will depend. And this is where it becomes very interdisciplinary, because there are people who will be probably researching these for more of a health promoting. Mm-hmm. For instance, if we think of kombucha, there's actually a lot of people who've been researching what it is about them, what is the small chemistry about them that is actually health benefiting, and if it's health benefiting or not. But yes, and yeah, so it's probably it's related. Like I said, there's many people who have interest in natural products.
Yeah. But would you say that the most of the work is happening and has happened in the let's beat the bugs or let's beat the cancer kind of mode?
Well, this is, okay. So I guess I actually think, so unfortunately, I think that this is where a lot of focus has been in for our research and development dollars.
Yeah.
So unfortunately, when you get more into the side of natural products where you might find them in health food stores, because all of those things that you find in health food stores are going to be the chemistry that's going to give that effect, there tends not to be the necessary research and development put into that, which there is actually numbers. So for instance, pharmaceuticals, and yes, there's been a lot of evil pharmaceutical companies where they've done some not great things, like in terms of our opioids and things like that. But at the same time, they put in 20% to 30% of their profits back into research and development. Whereas when you go towards more of the nutraceutical development, tends to be on the order of 2 to 3%.
Yeah.
So, and dollar-wise, they're probably relatively equal in terms of market share. So, but yet I think it's really complex because sometimes people, like if we're eating things as foods, there's a lot more to it than just the health benefits. There's a lot. There's a lot that goes into it that can maybe be hard to put I don't know, put a stamp on. But
That's interesting. Thank you.
Yeah.
How much does the diversity of the secondary metabolites sort of change from individual to individual, being like individual bacteria to individual bacteria, from species to species, and even from like generation to generation? Is the sort of turnover of secondary metabolites really fast?
That's a really good question. And it's actually, I think, a question that hasn't been answered. So we know the oldest streptomycin is a natural product that they've actually been able to sort of timestamp where it was first produced, like its gene cluster wise, that using bioinformatics, and was about 230 million years ago. So we know that's probably how old they are. We don't know how, like that actually is one question I'm really interested in, how fast, because even if there's a mutation in a gene cluster or a gene, it might cut off the final structure, but it might produce a structure that doesn't have any use for the organism that's producing it, but it might have use for us. Yeah, I think it's a question that hasn't been answered, and probably we have the tools now to answer it.
So when making semi-synthetic derivatives of natural products, is the way you alter the structure just kind of a shot in the dark, or is there something that gives you a clue of what ways to alter the structure, or how do you go about it?
So in modern days, you would probably model it. So, right, there'd be modeling programs where you'd put it in with your target and see which ways that you can get your optimal activity. My supervisor, been at it for a long time, would tend to look at a structure based on his background of knowledge and be like, well, we should try these things. Just, it's like that. He's the modeling in his head. So, and now he'd probably model things.
Are there any of those potential antifungal specialized metabolites that are like undergoing clinical trials that people think are really promising?
I have not looked that up recently, but I should. Yes.
Yeah, I'm just curious because I know there is this kind of big awareness just and whatnot. Just a lot of initiatives that are trying to mitigate that. So
yeah, oh, that's a good question.
Yeah, cool.
Yeah.
No worries.
All right. Well, thank you all for joining us. I really like hearing the history of how these products came into being and into pharmaceuticals, coming from neuroscience. A lot of the reagents people used in the labs were from spiders and snake venom. So I'd like to hear other stories about maybe less threatening species that we can work with. But thanks everybody for joining us in the room and online, and thank me again, or join me again in thanking Kalindi for presenting here today.